Welcome to CE on ReachMD. This activity is provided by Prova Education and is part of our Minute CE curriculum. Prior to beginning the activity, please be sure to review the faculty and commercial support disclosure statements, as well as the learning objectives.
This is CE on ReachMD, and I'm Dr. Lauren Byers. Today, I'll provide a brief overview of the rationale for targeting B7-H3 in extensive stage small cell lung cancer. So to start with, what is B7-H3? B7-H3 is the transmembrane protein which is overexpressed in a variety of cancers, including lung cancer, prostate, and esophageal cancers. And B7-H3 expression has been associated with poor prognosis. A finding from our group and colleagues is that B7-H3 is consistently expressed, as had been shown previously, across small cell lung cancers. And this is very relevant because we know with lung cancers that there can be differences in terms of target expression. But with B7-H3, we see it consistently expressed across all of the different types of small cell lung cancer. If we look at the mechanism of action for the antibody-drug conjugates, there are classical ADC modes of action, and this includes the ADC binding to the cancer cell and then bringing in this toxic payload, and this delivers a cytotoxic effect with the drug payload. In addition to the direct effect on the cancer cells, though, there are also advantages to this approach because of things such as bystander killing effect. And this includes mechanisms where release of the drug's payload from the antibody before being internalized can provide further therapeutic benefit for surrounding cancer cells in that area. And specifically, this is when the payload is being released into the intercellular space because of high drug membrane permeability. The drug payload is released after the linker is cleaved. And so again, this delivers a very high concentration, but specifically to the areas where we would want to deliver treatment. And this is a result of having a high drug to antibody ratio. These are an example of 4 B7-H3 targeting antibody drug conjugates that are currently in development. This includes IDXD, YL201, HS20093, and MHB088C, and several of these had updates that were presented at the World Lung Conference. A few things to really highlight here. These are all using topoisomerase payloads, and so similar payloads in terms of the killing effect from that payload. You can see that there are a range of drug to antibody ratio, with the highest being 8-1, but more of the other therapeutics using a 4-1 ratio. There are also some differences in these different therapeutics between the linkers, and so those may have some differences in terms of the activity of the drug. And what you can see also is that at least 3 of the 4 therapeutics that are being developed and that are shown here have bystander killing effect. I wanted to, again, highlight 2 of the therapeutics targeting B7-H3 that were presented in the antibody-drug conjugate session at World Lung. And again, this is QLC 5508 and IDXD. And I think what we are seeing in these programs and in the data that was presented at World Lung, that there's not only a very encouraging signal in terms of the response rates that are being seen in patients with relapsed small cell lung cancer, but also frequently patients are getting relatively fast response, which can translate into improvement in symptoms that they may have related to a cancer recurrence. Both of these that are highlighted here have phase 3 trials that will further let us understand how these compare to standards of care that are currently in use. Well, my time is up, and I hope you found this overview useful. Thanks for listening.
You have been listening to CE on ReachMD. This activity is provided by Prova Education and is part of our Minute CE curriculum. To receive your free CE credit or to download this activity, go to reachmd.com/CME. Thank you for listening.